the risks and the costs, the costs and the benefits of animal research. So Darwin went on to, after he did his book, The Descent of Man, where he said the difference in mind between man and higher animals, great as it is, certainly is one of degree and not one of kind. He recognized that we are on a continuum with animals, both physiologically and emotionally and psychologically. And he asserted in his book, and if you haven't read it, I encourage you to read it, The Expressions of Emotions of Man and Animals, where he was really one of the first persons that I know of who looked at the expressions of animals and attributed different emotions to them. So he could look at a dog and say, this dog is frightened, this dog is experiencing happiness or experiencing sadness. So he really was a pioneer in the field in that sense, recognizing that it was a dichotomy. And unfortunately, he couldn't do very much about it. And just as we're faced with that situation now, we really can't do too much about it. We ask ourselves the ethical question, so if we stop doing research on animals, what happens to human health? Where's the next antibiotic going to come from? How is the next surgical procedure going to be developed? And are we willing to sacrifice that because we say we don't want to use animals in research? So it's a difficult question. So now we know, of course, since Darwin's time, there's a lot of advances that have been made in ethology to show that some animals are capable of all of these characteristics, any or all of these characteristics. And of course, we know that some animals like chimps and elephants and dolphins have indicate self-awareness, facial recognition. Some of the work by Frans de Waal on primates, great apes, particularly chimps and bonobos, have shown that they naturally have aggression toward one another and they show altruism toward each other. Dolphins have language that, and other animals too, have language that they speak to each other. And so we're the ones who are dumb in the sense that we can't read our animals yet. And that's still an ongoing process. So given the fact that we feel in society has made the decision that we still need to use animals in research, what do we have to do to protect the animals? Well, I'm going to go through a little bit about the history of animal protection legislation in the U.S. and I'd point out that while in the previous slides I showed you that they were passing the Animal Cruelty Act in England in 1876, it wasn't until 1966 in the U.S. that the Laboratory Animal Welfare Act was passed that was really focused on dealers to prevent pet theft. And it included dogs, cats, non-human primates, guinea pigs, and rabbits. And at that time, there were several notable cases where pets had been stolen from their owners' backyards and sold into research laboratories. And so there was a big expose in Life magazine and several others that prompted Congress to finally act. And the person who was active in that area was a woman by the name of Christine Stevens. Her father was a physiologist. She began to question the way animals were used in research and started the Animal Welfare Institute. And it was through her efforts in going to Congress that persuaded them finally, and then on top of that, the stolen dogs, that finally persuaded them to pass the Animal Welfare Act. So the act has been amended a number of times. In 1970, it was expanded to include all warm-blooded animals, except farm animals. In 71, USDA made an arbitrary decision to exclude rats, mice, and birds from the act because of the sheer numbers of rats and mice that were being used and the impossibility of the USDA, which is the agency tasked to enforce the Animal Welfare Act, to deal with that number of animals. In 1985, there were probably the biggest, most notable changes to the Act, where institutional animal care and use committees were created. The law mandated that pain and distress in the live animals be minimized. They specified that dogs needed to have certain exercise requirements, and then they established guidelines to maintain psychological well-being in primates. So that was a big step in looking at the welfare of animals in laboratories. Now, I have to say that in the 50s, there was a group of animal scientists and veterinarians who had recognized the fact that a lot of the animals that were used in research were not necessarily in the best of health. So they had many microbiological diseases and carrying parasites and things like that. And so in the 1950s, when ILAR was started, it was really talking about trying to set standards for the physical quality of the animals, recognizing that if animals were not healthy, they're not going to be good research models. And it's taken up until at least 1985, and probably even it hasn't gotten there yet, to recognize that if laboratory animals don't experience appropriate psychological well-being and their behavioral needs aren't being met, then they're going to be in a state of stress and distress and not be very good biological models. So then, besides Animal Welfare Act that's overseen by the U.S. Department of Agriculture, PHS, or Public Health Service Policy, also applies to research in institutions that have funding from Public Health Service. And in this case, it protects all vertebrate animals, although Public Health Service Policy does not require institutions to count the numbers and report the numbers of animals that they use. So we still don't have an idea how many rats and mice or birds are being used in research, where you can go to the USDA website and find out how many of the other species are being used on a yearly basis in the U.S. You can only guess on the numbers of rodents that are used. Institutions have to apply the guidelines set forth in the Guide to the Care and Use of Laboratory Animals, which, as Alicia mentioned in the introduction, ILAR published the latest edition of that in 2011. And then each facility has to have an ILAR report whether or not they're accredited by ALAR International. And then besides those two sets of policy, we also have the Association for Assessment and Accreditation of Laboratory Animal Care International, ALAC. And I'm sure that many of you have heard of that. You know, Tufts is an ALAC accredited institution that was brought on by one of the former deans, Frank Lowe, who I guess lives in perpetuity on this campus and this facility. He was a wonderful man. I actually shared him. He was at Hopkins for quite a while, and I knew him not well, but I knew him. He was a wonderful man. I did a lot for animals too. Anyway, getting back to ALAC, this is a voluntary accreditation program. It uses the guide also, but then they also use some other guidelines since they are now international. And they do announce site visits every 3 years. So when you hear that the facility is preparing for ALAC site visits, it's probably going to be a lot of frenzy and weeping and gnashing of teeth as people try to get things in order for the site visit. So that's kind of the policy in sum and in a nutshell. I did want to highlight that there have been a couple of recent changes to NIH policy with regard to laboratory animals. Both of them came about from reports that were released by the National Academies. The report on Class B dealers that was done when I was still there, where NIH now has prohibited NIH-funded researchers from obtaining drugs and fats from Class B dealers. Everybody know what a Class B dealer is? Okay. Class B dealers are dealers that do not breed animals themselves. They go to places, they find them, they buy them, they keep them, and then they sell them to somebody else. And so many Class B dealers have been shut down because of the atrocities and the way they treated their animals. And then there was also some question about where they got the animals. Now, the chance that they actually got them from private owners is pretty small, but they didn't have, there was no paper trail for certain numbers of those animals, so it's never really been proven that animals were not stolen. pet animals were not stolen, but mainly because of the conditions under which the animals are kept. These guys they were pretty shady characters keeping these animals, and there was an expose that was done, I think it was either on Discovery or the Animal Planet, of a place in Texas or Arkansas, a Class B dealer that was exposed by one of the animal protection groups, and it was horrifying. And when the committee that wrote this report saw that, they just were incredulous that this was really going on, and that the NIH allowed people to buy animals from those kinds of places. So that's the good news. I think that's a really good thing that's happened. And now, because there are certain types of animals that were obtained from random source dealers, like deep-chested dogs and things like that, the NIH is now working with Class A dealers or actual purpose dealers, purpose-bred dealers, to breed mixed breeds or hounds and that kind of thing that are used for very specialized types of research where Class B dogs were used in the past. The other thing, and I'm sure you've heard this, is that most of the use of chimpanzees for biomedical research was deemed unnecessary by a committee formed by the Institute of Medicine. And any proposed future research on chimps must meet certain strict criteria. So you can't just do a standard IACUC proposal and saying I want to do research on chimps. It has to meet certain criteria. And most of the chimps that are owned by NIH, particularly there are only 700 chimps in the U.S. that are owned by NIH, and most of them will be retired to sanctuaries where they will be allowed to live out their normal lifespans in nice conditions. So that's, I thought I would just mention that because policy is always changing, and it's always possible to get things done. It's just a matter of really keeping at it and being persistent in really understanding the science behind it. I think that goes a long way to convincing the Congress and the research community that things are science-based and show evidence for something. So if animals are not well kept and they're diseased and they're being sold for research, that's not very good scientifically. So you have to play the science card a lot of times when you're trying to get, when the animal welfare card goes over, okay, but it's not going to convince anybody. Well, not many people, I should say. So the guiding principles for the use of animals in research, I mentioned earlier, the 3 R's, replacement, reduction, and refinement. These were principles that were established in a book that was published in 1959 by 2 British scientists, Russell and Birch. And they were tasked by the University's Federation for Animal Welfare to go to all the research facilities in England and look at the conditions under which the animals were being kept and make some recommendations for improving the welfare of animals and improving the science. And again, Russell and Birch approached this very much from a scientific background. They were both scientists. And so when they published their book in 1959, it was accepted by UFAR, or the University's Federation for Animal Welfare, but it didn't really make the headlines. And so much of what was published in this book really went unnoticed until the 90s when Martin Stevens from the Humane Society of the United States actually dug it up and went and literally found Russell and Birch, who was still alive in the UK. And we had a meeting in 1995 with them on talking about the future of the 3 R's. And I had the privilege of meeting them. They're both deceased since then. But and what they really meant when they did the work for this book. So their first principle, the way they present it, and you won't always hear it presented in this order, but I forgot that I have this quote in here, but this is from the book. By now, it's widely recognized that the most humane possible treatment of experimental animals, far from being an obstacle, is actually a prerequisite for successful animal experiments. That really set the tone for the book. So in talking about replacements, we talk about living systems, organs, tissues, or cell culture techniques or in vitro techniques where animals don't have to be used, so you can completely replace animals. Or use less sentient animals, so moving from using non-human primates to maybe using zebrafish, for example. Invertebrate models using worms and flies and that kind of thing, microorganisms, of course. It's also possible to use non-living systems, so just plain old chemical and physical reactions to test for corrosivity of chemicals and things like that. No need to put it on an animal's skin if you can test it. And then now probably one of the most advanced ways of doing it is by developing computer models, and which we call in silico systems. So a lot can be done without animals, at least at certain points in the investigations. Then the next step, if you can't replace, then what do? You reduce. So these are some of the considerations that they talked about for reduction of animals. Rational selection of group size, including pilot studies, and power analysis. So in other words, figure out before you do your experiment how many animals you're going to need to get statistically significant data, rather than afterwards and try to find a statistical method that will fit your data. Careful experimental design, which goes into the same principle. Maximizing the use of animals, so that for each animal that you have, you get as much data as you can from each animal. Choosing the correct model. You know, so many times in science, models are used because of for historical reasons. So everybody's always done this research in mice, so I'm going to continue to do it in mice, even if mice aren't the best model. So I think we're coming to a very critical time in biomedical research where people are really beginning to think about the value and validity of animal models, that they may be valid for one thing but not for another. They may be valid for a piece of one disease but not for the entire progression of a disease. And it really requires a lot more dissection of the model. And I'm going to get into that a little bit when we talk about the toxicity testing pathways and how that might play into this. And then, of course, we talk about optimal animal husbandry. I mentioned these before, monitoring for infections, avoiding overbreeding, so you don't have so many excess animals to euthanize. And as we talked about, too, the minimization of distress, which usually comes under the refinement conditions. So we talk about refinement as maximizing animal well-being and minimizing pain and distress in any animal that is undergoing a procedure in the lab or just under normal husbandry conditions. So we look at pain, we look at distress, we look at humane endpoints or ending an experiment where you can still obtain the data that you need without letting an animal suffer right through to the end where it has to die. And many people are faced with that now because a lot of animal care and use committees will not allow death as an endpoint in animal studies. And I think that's a tremendous advance. It forces the investigators to think about where is the point of no return in this particular animal model once it loses 20% of its body weight, once its temperature goes to, if it's hypothermic for a given period of time, we know that animal will never recover. There's no point in allowing it to suffer through that. So killing the animal at an earlier time point for the main reasons. And then the whole concept of environmental enrichment, which deals with animal husbandry and meeting the behavioral needs of the animals in a laboratory environment, which, as you know, can be very difficult. So now I'm going to change gears a little bit and talk about toxicity testing. And you're going to say, what does this all have to do with it? Well, the Center for Alternatives to Animal Testing, where I work, was founded in 1981 because the animal rights movement put a lot of pressure on the cosmetics companies to stop testing cosmetics on animals. And Henry Spira, who is an animal rights activist based in New York, took out a full-page ad in the New York Times that said, how many rabbits does Revlon blind for beauty's sake? And Revlon then was kind of on the hot seat, because in order to test chemicals for eye irritation, you have to take an animal like a rabbit, and they have the most sensitive eyes, as you probably know. And you would put the test chemical in one eye and leave the other eye as a control, and then look and see what happened to the animal's eyes. And maybe for cosmetics, it might not be so bad, but when you start testing chemicals and other really nasty things, it's pretty painful for the animal, and it's very inhumane. So our center was started by the Cosmetics Trade Association to look for alternatives for testing cosmetics and other agents in animals, so that we don't have to use animals anymore. So that was really planted the seed for this whole movement to look at alternatives. So what is toxicology? That's really my scientific background, so I have to give you a little bit of this. Informally, as you know, it's the science of poisons. Paracelsus, who is the father of toxicology, came up with the wisdom that the dose makes the poison. So everything would be toxic at a certain level, but toxicology is in the business of determining where that level is, what is toxic. So pharmaceuticals, for example, most of them you can overdose on and kill yourself, but there is a dose that is effective. So toxicology looks at that, the adverse effects of chemicals and agents, including drugs, on people, animals, and the environment. So toxicity testing is really a systematic approach to assessing the toxic effects of chemicals or drugs on human health. And we used to have tests called the LD50. Anybody heard of the LD50? The LD50 is the dose that kills half the animals. And they figured, okay, once we know what dose kills half the animals, then we can make a regulatory decision on what is going to be a safe dose for humans. Pretty crude. You know, toxicology was always known for, you know, give an animal a chemical and then count the pause in the air after a couple of days to see how many died. So, you know, it was refined over the years so that not only are you measuring death as an endpoint, but you were measuring different pathologies, so you'd know what a chemical is actually doing to an animal. So why do we test? And I'll just go through this very quickly, but we have a lot of legislation that was put into place to protect human health. In 1938, the Federal Food, Drug, and Cosmetic Act was passed because people died from a mascara that actually caused an eye infection in a woman to die. It hadn't been tested on animals. There was an elixir of sulfanilamide that when it was mixed in the lab, they made it with diethylene glycol. As you know, that's very toxic. So it killed a number of children who took this. It was meant to be a sweet-tasting, you know, medicine for kids, and it ended up killing them. So Congress said, okay, that's it. We have to test. And so how did they mandate that things be tested? They're not going to test them on people. They mandated that they be tested on animals. And now the way the law stands is you have to test on one rodent and one non-rodent species for drugs. Then came FIFRO, the Federal Insecticide, Pesticide, and Rodenticide Act, because of all the pesticides that were put on crops and that were being ingested by humans, and kids, particularly kids, subjected to what might not harm an adult would certainly harm a child. So they mandated that pesticides be tested, again, on animals. That was done initially by the USDA, but when the Environmental Protection Agency was formed in 1970, they assumed responsibility for testing pesticides. And then the Toxic Substances Control Act dealt with all the chemicals that are in commerce. So all these big trucks that you see that are on the road that contain all these chemicals, and they say hazard on the side. Those are all chemicals that you have to be able to know what their toxic effects are. So if a spill occurs, we know how to deal with it. And then most recently, the endocrine disruptor screening program, so testing for agents in the environment that act as endocrine hormones that could be toxic for either humans or animals or the environment. So just a point to bring home here is that even though the Toxic Substances Control Act was passed in 1976, it did not give the EPA any authority to test chemicals. In fact, it allowed most of the chemicals that were already in commerce to continue to be sold unless somebody showed that they were harmful. And for new chemicals, it was really under the auspices of EPA to show that they were dangerous, not the companies who were synthesizing them. And a great example of how this system does not work is, how many of you remember the spill in the river in West Virginia just a few months ago? So it was a chemical called 4-methyl-cyclohexane methanol. They didn't have a material safety data sheet on that compound. They didn't know what it did. All they know is that it smelled like licorice in the water, and they said, don't drink the water. They had no idea. So you see, for public health reasons, we need to know about these chemicals. But if we started to test them all in animals under the protocols that are required to know all of the different types of organ toxicity, acute toxicity, reproductive tox, it would take millions of animals and probably thousands of years. It just can't be done. So from a practical reason, we need to find another way to look at the toxicity of chemicals. So traditional, and then the other problem with toxicity testing of chemicals is that most of us are not exposed to huge amounts of chemicals. We're exposed maybe to small amounts in drinking water or in food, but you can't test an animal with that because you'd never pick up any toxicity. So instead of testing the amounts that we're actually exposed to, they test massive quantities of these chemicals, massive amounts in animals, so that they can find out what kind of effect it's going to have. And then they have to extrapolate back to what will happen if you're exposed in low doses. And then they put in all these fudge factors to try to give levels for regulation purposes, what's a safe dose for humans. They use animals that are typically inbred strains to test these chemicals, and then they have to, again, extrapolate the results to an outbred human population. Another fudge factor. So you can see how inaccurate a lot of these methods are, but it's what's been done and what's been relied on. It's currently the law. So it's not surprising that the results from animal studies don't always reflect results in humans. And even so, if you look at the list of carcinogens in animals and you look at the list of known carcinogens in humans, they're very different, because the known carcinogens in humans that have been demonstrated from human evidence epidemiologic data is very small compared to the list that's given for likely carcinogenic in humans because it's carcinogenic in lab animals. But you know, you remember those studies, I guess I'm dating myself, but they were reporting studies on saccharin where you had to give a rat the equivalent of, I don't know, 3 pounds of saccharin a day to see a tumor. And as it turns out, rats have a very special metabolism in their kidney that they develop kidney tumors, but it doesn't happen in humans. So the size of the dose, the animal model, most of these things are not relevant. They're the best we have at the moment, but they're not really relevant. So again, another Academy report in 2007, the EPA requested the Academy to do a report that was titled Toxicity Testing in the 21st Century. Where are we going with this? Our science is not back in the 19th century or even the early 20th century. We're advancing, we're learning new things. What can we do to improve our testing strategies? So basically what the committee decided, they wanted to see a paradigm shift so that we could provide broad coverage of chemicals, chemical mixtures, which are totally enigma to the testing community, and outcomes and life stages. So we don't do testing on young animals or old animals. We do testing on this very standard group of animals. So we don't know how to extrapolate that to life stages. We need to reduce the cost and time of testing. We need to use fewer animals and reduce and cause minimal suffering in the animals that are used. And then we have to have a more robust scientific basis for assessing the health effects of these agents. So all of those are really great ideas, but getting them into practice is another story. So this was, I'm not going to go through this, but all I'm going to do is show you that this was printed in the report. So option 1, the in vivo model, which has been the traditional model using animal biology in high doses and low throughput, expensive, time-consuming, using a relatively large number of animals in very apical endpoints, not based on any kind of mechanism at all. Where the gold standard, option 4, would be to be doing everything in vitro, so, and using human cell systems, human biology, with a broad range of doses, high throughput, less expensive, less time consuming, almost no animals, where there may be times that we would need to go into animals for certain things. And instead of looking at apical endpoints, we would look at perturbations of toxicity pathways. So understanding how the chemical works on a molecular level to then predict what's going to happen at an organismal level. I'm not going to go through this. Well, I'll go through it very quickly. But so looking at what is a toxicity pathway. So you have a biological input and a biological function. So in the old days, treat the animal with a toxic chemical, animal dead. So here, if you're talking about low dose, you look at the tissue dose of the chemical, you look at the biological interaction and the perturbation of homeostasis. You see early cellular changes. But we do have the capacity to adapt. So at a low enough dose, we can adapt stress responses and go back to normal homeostasis. In a case where you have a higher dose, now we're going to not only see early cellular changes, but we're actually going to see cell injury and perhaps morbidity and mortality. So when the body is no longer able to adapt to the insult from the chemical, then you will have a more serious effect. So essentially, the way we're going about doing this is looking at toxicity pathways. Where does a chemical affect a pathway, normal biological pathway in the body, and be able to then predict what will happen to the organism based on that. Now, as you all know, this is really in its infancy, and I wish I could give you data on it, but that's really where the field is going at the moment. And there are a lot of people that are working in this area. So how does this impact policy? Well, here we are again with the National Toxicology Program then published a vision and a roadmap on how to improve toxicity testing for its programs. And then as a result of the National Academy publication, there was a memorandum of understanding that was developed between the US EPA, National Toxicology Program, NIH, and the FDA to incorporate new testing strategies into toxicology. The EPA, the FDA, and the NIH administrators all agreed to sign on to this as a worthy cause to have their agencies get behind and improve the methodologies. And then I wanted to mention the ToxCast program that's being used at the EPA, where they're using automated chemical screening technologies called high-throughput screening assays to expose living cells or isolated proteins to chemicals. In their first phase, they did over 300 chemicals in 500 different assays. Typo there. And then they screened it for the second phase, screened over 2,000 chemicals from a broad range of sources, including consumer products, industrial chemicals, drugs that never made it to market, 700 assays for over 300 toxicity pathways. And this is all done in an automated fashion. So you see the robotics there. They have these 1,500 well plates. They have chemical exposure to all different types of human cells, looking at different endpoints. In this case, they're looking at estrogen receptor response, and then trying to pinpoint where in a pathway these chemicals might affect and cause a toxicity. So it's a screening program. It's not perfect. They're looking at single points in time in various, very specific cell types. But it's providing a lot of basic information that we never had before. We have omics technologies that I'm sure you're all aware of, genomics, transcriptomics, metabolomics, proteomics, to try to identify what genes are being affected by toxicants, what proteins are being affected. How can we put this into a pathway-based approach to understanding how a chemical is acting? Probably one of the most exciting things I've seen recently is the organ on a chip. Has anybody heard about that? Yeah. It's fascinating. I mean, the group over at the Vivis Institute at Harvard is really developing a lot of it. And most of it is supported by FDA, NIH, and the defense industry because of needing ways to screen for bioterrorism, anti-bioterrorism agents, which you can't really do in human. You can't do clinical trials on drugs that are going to interfere with bioterrorism agents. It's pretty unethical to do that. So they have to, and the animal models are not accurate enough to predict what will happen in a human. So what do you have to do? You have to rely on a human system as much as possible. So you may have seen this before if you've heard about it, but this is absolutely fascinating. They take human lung cells and put them on one side of a chip. They put capillary cells mimicking the blood vessel on the other side of the chip. They have blood flow through and IL-2, and they can look at effects of different chemical agents from the exposed through the blood. And then they have airflow coming through here, through the lung cells. And then on either side, they have a vacuum that puts stress on the lung cells, similar to the stress that we provide our lung cells when we breathe in and out. Because our lung cells don't sit there static condition. We breathe and they expand, and we exhale and they go back. They found, looking at the biological effects in this kind of a system, if they did it without the physical constraints and putting the stress on the system, the cells didn't respond. When they did put the stress on the system, the cells responded exactly as they would respond in a human lung, which is pretty fascinating. I mean, every time I see this, it just drives me into, this is the future. This is where things are going. And so not only are we doing that, but they're working on liver on a chip, heart on a chip, kidney on a chip, bone marrow. I'm aware that we're getting close on time. Do we have to wrap it up? Okay. Yeah. So I didn't get a chance to go into where the future of testing, in my view, is human on a chip, that someday we will be able to integrate all of the organs, human organs, so that we are able to decipher some of the mechanisms in vitro systems. We might not be able to do everything, and we might not be able to do it all rapidly, but I think that in the long run, we will get a lot of answers out of it. Neuroscience is a kind of a field that's going to present a much different challenge because it's very difficult to model human brain and brain function and how we behave. But anyway, so I just want to skip through the last few of these. I'm going to make you aware that most of our lab mice are kept under hypothermic conditions and they're constantly under stress. So some work that's been done in the refinement area by Joe Garner and Brianna Gaskell showed that if you put between 6 and 10 grams of nesting material in the mouse cages, they're able to make nests and thermoregulate. Otherwise, they're thermostressed. So that's an important take-home message. Environmental enrichment, the idea of providing novel stimulus in the animals in their laboratory environments to encourage species-specific behaviors and to avoid the stress and stereotypical behaviors resulting from boredom or fear. That's where things are going in that field. Group housing, looking at group housing in social species and how to accomplish that. In the new guide, that's the new norm now. It used to be house animals socially where possible. Now it's house animals socially unless for extreme experimental conditions you can't. So if an animal is a member of a social species, we must house them together. So things that, we have to look at the benefits of enrichment, so we can enhance species-specific behavior, but then we might cause aggression and wounding in animals that are not compatible, food competition, infant mortality. So you all know as veterinary people what some of the possibilities might be and some of the difficulties might be, but if you don't socially house animals that are social, there are also consequences. And these are just, and you'll have these slides available, but I'm not going to go through this, some of what's being done now for non-human primates and for dogs and for other animals. And then the other thing I wanted to just briefly mention is the positive reinforcement training, which I'm sure you're all aware of, that a lot of stuff has been done in primates to train animals to cooperate in experimental procedures. And the work that's been done by Molly Bloom-Smith and Steve Shapiro and others has just been remarkable in the way they're using positive reinforcement training to get animals to cooperate in just normal husbandry type things as well as in experimental procedures toward the goal of making the animals a willing participant. They're still not able to give consent, but at least if they're willingly participating, it's a step in the right direction. So I'm just going to say that I do see that someday we will not use animals in research. It's not an if, but a when. And I think people will disagree on when the when is. I'd like to see it happen in the next 20 years, but I don't know if that's possible. It's very difficult to predict. It depends on how fast science progresses. And the better the I think that probably one of the best examples of this is human on a chip being developed because of Department of Defense needs, right? So they're pouring all of this money into developing human on a chip. If they decided that they wanted to find, the agencies, the powers that be that hand out the money, if they decided they want to find better methods that don't use animals and they put some money behind it, you'd find the scientific community getting right behind it and doing it. And it could happen quickly because the science is really developing fast. But until that time, I think we just need to continue to employ the principles of the 3 R's. And that's the best we have right now, the best system of guidelines that we have, and the best metric that we have for ensuring that our animals are the best models from a scientific perspective and that we are treating them in the best possible way that we can. So I will stop there and show you my menagerie, our new puppy. Milton, he's a real joy. I haven't had a puppy in 30 years, so it's, and our 2 black blob cats. So, but thank you for your attention, and I'll be happy to take questions if we have a few minutes. We have a couple minutes before we have to let the other time. Anybody have any questions? Yeah. That's a great question, and I'm glad you asked it because I didn't put it in there, and I should have. The question was that there are 30,000 or more animals that are seen in the hospital here at the veterinary school with naturally occurring diseases. Why can't we use them for clinical trials? And I think that's the way things are going. I mean, that's something that I'm trying to, we're trying to get started at Hopkins now because we have an incredible imaging facility that could easily, it's used for laboratory animals, but it could also be used for client-owned animals in the Baltimore area. But then also we could partner with veterinary schools in the technologies that we have available to us at Hopkins, and we could do clinical trials in companion animals. And I think that would give us a remarkable set of data to work with, because, yeah, no, I think that's the future. You know, in order to get NIH and FDA on board, I think it's a matter of just continuing to press the issue with them and to continue to go forward with applications, grant applications. You know, the veterinary community has not done well strategically with NIH funding, which is really too bad. But to be honest, you know, the NIH has a huge comparative oncology group that you're probably aware of, that they're doing work on site, on campus, where they are using companion animals, so they know about those things. Why they're not funding it, I have no idea. I mean, I think that it's a tremendous resource that's not being taken advantage of. And the other thing is the epidemiologic data that we could tap into from private veterinary practice, because veterinarians all over the country are seeing animals with all of these diseases, and we're not taking advantage of that knowledge and what we can be doing about that. So I think that really is the next step, and the whole One Health initiative is, you know, it was there back in 2008 when it was first initiated, but I don't think it's taken off in the way it should be taking off. So I totally agree with you, and whatever we can do to emphasize that can't be enough. Yep.